I'm pressing the, oh, here we go, okay. So, gradual Darwinian evolution by natural selection actually paradoxically predicts that punctuated speciation dynamics in cases of primary and secondary contact. And as populations actually slowly accumulate adaptive differences at many loci with sort of modest to low effects on fitness, you can reach a tipping point. And at this tipping point, individual genetic effects of selected loci actually can become coupled to reduce gene flow genome-wide, facilitating their sort of rapid collective divergence. And thus, at these tipping points, actually small changes can have really rather large impact for increasing reproductive isolation and for speciation. So what I want to see today is whether there's any evidence for such tipping points and the genome increasingly sort of becoming the unit of divergent selection in the transition from host plant associated races to sibling species of Rhagoletis palmella flies. And when we talk about these punctuated divergence patterns, we're actually referring to sites affected by selection. So to sort of concentrate or focus on these sites, we looked at eclosion time differences, which is a key trait for these flies, adapting them to differences when their host plants fruit. And our strategy here is to do a genome-wide association study to ID these selected loci or selected SNPs. And what we do is then rad-seek early and late eclosing hawthorn flies in this case, and see if the differences in allele frequencies between early and late eclosing flies, hawthorn flies, predicts the pattern of genetic differentiation you see at these SNPs out in nature between the races and the sibling species. And when we do this, we actually find that eclosion time differences for 10,000 SNPs actually predict genome-wide patterns of genetic differentiation between the host races. This is the apple and haw host races, for Rhagoletis palmella out in nature. And that when we look at the sibling species, in this case the dogwood fly, the prediction is even steeper. So essentially what we see is that species differ in the degree of differentiation, but not in kind from the races, showing sort of more pronounced frequency differences for eclosion time related SNPs. And also what we see is a general emergence of significant linkage disequilibrium between eclosion time SNPs mapping to different chromosomes between the species, but not the races. So this results in the genotypic clustering of individuals at the local level between sympatric populations of species, but not races. And also what we begin to see is the global clustering of different host-associated populations of species, but not races, when we look sort of globally across the geographic landscape. So thus, it appears that the genetic differentiation, while similar in type, is organized differently, and the genome seems to be increasingly acting as a unit of selection as we move across that transition from races to species of Rhagoletis flies. And then the question then is, do other systems of primary and secondary contact show similar sorts of tipping points and, if you would, punctuated disequilibrium patterns of divergence with gene coupling and congealing during speciation? Thanks.